Welcome to today's lecture on molecular biology. In this session, we will delve into the fascinating world of gene expression and translation. These processes are essential for understanding how genetic information stored in DNA is converted into functional proteins. So, let's get started. The central dogma of molecular biology is a fundamental concept that describes the flow of biological information. It states that genetic information in living organisms flows in one direction, from DNA to RNA to proteins. This concept was first introduced by Francis Crick in 1958 and has been widely accepted ever since. The genetic code is a set of rules that determines how the nucleotide sequence of DNA or RNA is translated into the amino acid sequence of a protein. This code is universal, meaning that it is shared by all living organisms. It consists of combinations of three nucleotides, known as codons, which encode specific amino acids or serve as stop signals. Gene expression refers to the process by which the information stored in a gene is used to create a functional product, such as a protein. It involves two main steps: transcription and translation. Transcription occurs in the nucleus, where a DNA sequence is transcribed into a complementary RNA molecule called messenger RNA, mRNA. The mRNA then travels to the cytoplasm for translation. mRNA is a single-stranded molecule that carries the genetic information from the DNA template to the ribosomes where it is translated into a protein. It is composed of a series of nucleotides, including adenine, A, cytosine, C, guanine, G, and uracil, U. The specific sequence of these nucleotides determines the sequence of amino acids in the resulting protein. The ribosome is a complex molecular machine responsible for protein synthesis. In bacteria, ribosomes are composed of two subunits, the small subunit, 30S, and the large subunit, 50S. The small subunit is responsible for decoding the mRNA, while the large subunit catalyzes the formation of peptide bonds between amino acids. Translation initiation is the process by which ribosomes assemble on the mRNA to start protein synthesis. In bacteria, this process involves several initiation factors, including IF1, IF2, and IF3. These factors ensure the correct positioning of the ribosome on the mRNA and the selection of the correct start codon. The Shine-Dalgarno (SD) sequence is a short nucleotide sequence located in the mRNA upstream of the start codon in bacterial cells. It plays a crucial role in translation initiation by interacting with the complementary sequence in the small subunit of the ribosome. This interaction helps in positioning the ribosome correctly and ensures efficient translation. The start codon is the first codon in the mRNA that signals the initiation of protein synthesis. In bacteria, the start codon is usually August, which codes for the amino acid methionine. Recognition of the start codon by the ribosome involves the initiation factor IF2 and the initiator tRNA loaded with methionine. After the recognition of the start codon, the ribosome assembles into an initiation complex. This complex consists of the small subunit of the ribosome, mRNA, and initiator tRNA. The assembly process requires the participation of various initiation factors and GTP, a high-energy molecule that provides energy for the process. During translation elongation, the ribosome moves along the mRNA and adds amino acids to the growing polypeptide chain. The peptidyl transferase center, located in the large subunit of the ribosome, catalyzes the formation of peptide bonds between adjacent amino acids. The A site, or aminoacyl tRNA site, is where incoming aminoacyl tRNA molecules bind to the ribosome. Translation elongation is facilitated by various elongation factors, including F2, FTs, and FG. These factors help in the accurate positioning of incoming aminoacyl tRNA molecules, the translocation of the ribosome along the mRNA, and the proofreading of codon-anticodon interactions. Termination of translation occurs when the ribosome encounters a stop codon in the mRNA. Stop codons, WA, WOG, and UGA, do not code for any amino acids but instead signal the termination of protein synthesis. Termination factors, such as RF1 and RF2, recognize the stop codon and catalyze the release of the synthesized protein from the ribosome. In conclusion, understanding the molecular mechanisms underlying gene expression and translation is crucial for unraveling the complexities of cellular processes. The central dogma of molecular biology provides a framework for studying these processes, while the intricate interplay between mRNA, ribosomes, initiation factors, elongation factors, and termination factors ensures accurate and efficient protein synthesis. We hope this lecture has provided you with a comprehensive overview of these topics and their significance in the field of molecular biology.